opportunity for people to ask tons of questions. So just a reminder, if you do have any questions, to put them into the chat box, and then we'll get to those after the end of the 3 presentations. So next we're going to turn to Dr. Philipp Schatz, who's going to talk about the new launch of this gold standard in quality control biospecimen studies. Over to you, Dr. Schatz.
Thanks a lot for this nice introduction, and thanks as well to Dr. Philipp and Dr. Zapoka for those brilliant presentations which highlighted the importance of high quality control and the impact of very important clinical features such as dopamine and several vitamins which we have just seen. Now I'd like to introduce you to a solution to this pressing issue of bad quality samples in biomarker research, and this is provided by a product which we have launched at spring this year in London at the Biobank Global Congress and in Orlando at the ISPO annual meeting in Florida, and it's called MXP Quality Control Plasma. And this helps to upgrade existing quality assurance to a phase in which we can truly quality control the sample, not relying on documentation, but really understanding what the impact of the individual in the individual sample is to the overall quality of the specimen. I'd like to start with the first example of our internal research, and here we have analyzed samples derived from a clinical trial which used ongoing sampling of at the very same clinical site over a longer period of time. And what we've done is that we have looked at a certain ratio of 2 different quality measures, and we've looked at that over the time, and what you can see is that at a certain point in time, we suddenly observe a subgroup which suffered from insufficient quality, and this was true for both individuals suffering from a disease and those which do not suffer from disease, meaning that this was an independent measure, independent of the overall health status of the sample, however, highlighting the importance of continuously quality control, enabling additional training at that site, and potentially excluding those samples from downstream analysis. We do the assessment of the quality of the sample in 3 different measures. So first of all, we analyze the used plasma or blood collection tube in the first place, being able to distinguish between EDTA plasma, heparin plasma serum. Second, we assess the time prior centrifugation, so whether or not the time between blood draw and separation of the blood cells has been delayed. And at the third place, we analyze as well the storage of the plasma sample itself, and whether the separation of the blood cells has been done according to standard operating procedures. This all translates at the end into one single quality score, which provides you with a comprehensive overview of the quality of a given individual sample. We have beta tested this protocol together with a company which most of you should know, that which is Bayer Pharma, and they donated us or provided us with sample material blinded to us, suffering from different confounding factors. In the first place, they selected different anticoagulant compounds such as EDTA, citrate, and heparin, and with just 3 different analytes, we were able to clearly distinguish each of those individual coagulant groups, and were able to identify those samples which truly used EDTA plasma blood collection tubes in the first place from all others. In addition to that, we have as well introduced other confounding factors, such as the delay of the blood prior centrifugation and the longer incubation of plasma material after separation of the blood cells. And last but not least as well, the introduction of additional freeze and thaw cycles to the plasma samples. What we've seen is that our test was capable of identifying correctly 80 out of a total of 20 samples not suffering from any confounding factors as truly being of high quality, translating to a specificity of 90%. And while analyzing confounded samples, we were able to identify 98 out of 100 samples, translating into a sensitivity of the same assay of 98%. The assay works very easy. The starting material is, and this is true for all assessments which I've already introduced you, so everything is based on a single plasma sample, which is then further processed is very easy chemically, including an extraction cleanup step and a derivatization step in order to be able to analyze the sample with GCMS. The analytes are afterwards analyzed with the algorithm integrated into a software which is provided by us into a data or sample quality report, which allows the assessment of all those different aspects which we have already learned to. Such a protocol looks as it is highlighted on the right side of the slide. It's composed in a way that at the top of the report we see an overview about the entity of all samples analyzed in one batch, and below that we have individual assessments of different samples, providing each and every time 3 different informations: the identity of the used blood collection tube, the assessment of the blood processing control, and the assessment of the plasma processing control. We use a traffic light system to color code the quality of the sample, and this is provided for each different assessment individually, which can be explained as follows. The blood processing control, if it's highlighted as a green sample, the blood cells have been separated within 2 hours after initial blood draw. If it's medium quality, it's between 2 and 6 hours, and if it's above 6 hours, then the sample is highlighted as a red sample, meaning that it suffers from insufficient quality of the blood processing control. A similar system is provided for the plasma processing control, where we as well see that elongated time leads to a lower score, translating into a red color coding of the individual sample. However, this color coding provides you only with an overview which you can quickly assess. However, a more accurate scoring is given by individual numbers for each and every of those assessments, and as well translating into an overall quality score which provides you with a comprehensive scoring of the entire sample. I'd like to share with you now a second example from our internal research, and here we started with analyzing or with the discovery of a new biomarker which should be able to distinguish between cases and controls. However, in the first place, we see that we observed an insignificant p-value, meaning that the 2 groups were not able to separate from each other. In a subsequent quality assessment of the very same samples, we were able to identify a group, which is here highlighted in green, which had a very good quality, and a second group, which is highlighted in red, which suffer from low quality of the used sample material. Due to the information we got from our quality assessment, we were able to identify that those samples suffered from oxidative stress, and by excluding those samples from the data analysis, suddenly the difference between the 2 groups became apparent, and we generated a significant p-value. In other words, a study which would have been a deal breaker, so an entire stop of the discovery process, suddenly turned into a big success, and we were able to identify a new biomarker for that particular disease. The service we are here offering is actually available in two different manners. So we can either get samples from your facilities and analyze the samples in our laboratory and provide you with a report, or alternatively, we can train your staff in order to be able to run those analyses in your own laboratory, which of course would require a GCMS device. The price range ranges from 5K for a low amount of samples up to approximately 100 or below 100 euros per assessment per sample if it's performed in a service manner. For customers who are more interested in high throughput analysis and have a higher demand of quality assessments, we offer as well the licensing model, which comes with a much lower price depending on the amount of samples you analyze. The system here works very similar to a mobile phone contract. We provide you with the software and you purchase keys which can then, or which allow for a certain amount of analysis which comes with a certain price tag depending on the overall amount of samples you analyze. I'd like to finalize my talk with the different areas of application in which we believe this new service is valuable. So starting with the pharma and diagnostic industry, here we really provide a tool enabling the upgrading of the existing quality management towards a quality control, knowing what you're doing, and as well the monitoring of clinical trials ongoing in order to verify that the collection has been done correctly or if additional training is required. In addition to that, prior purchasing high valuable sample material, one could assess such a collection in order to justify investment. From a biobank perspective, ongoing monitoring is as well very valuable and can be used to certify that the internal protocols are very accurate and very reliable, and the derived quality of your samples are worth the price you're offering to your customer. In addition to that, obviously you have a tool in hand to train your own staff. Last but not least, from a clinical research organization point of view, again, you have here a tool in hand which enables you to prove that you are offering superior quality in comparison to your competitor, and enabling this not only in a single point, but ongoing as a monitoring tool in ongoing clinical trials. With that, I'd like to close and thank all the participants, and we are all three now ready to answer your questions.